Today I'm representing the Links Common Fund program. And also, as you may know, I'm also a PI of the newly developed CFDE Data Resource Center. So some part of this presentation will cover some of the activities that we are doing for this Data Resource Center for the CFDE. So the Links program was a common fund program that was for 10 years. It ended exactly 4 years ago. And we were the data coordination center for Links together with Stefan Schurer from the University of Miami and Mario Medvedovich from the University of Cincinnati. So a little bit about Links. So the concept of Links started with this, I guess, revolutionary seminal paper that came out from the Broad Institute, headed by Todd Golub, and the first author was Justin Lab. And I'm sure many of you heard about this concept of connectivity mapping. So the idea is to build a database where you have collected a massive amount of gene expression signatures for single drugs and other genetic or chemical perturbations. And then you can come in with your project, your biological interest. That can be a disease state versus normal, a mouse model, or some cells treated with some condition like a compound that you want to see what it does. And then you come into this Links database, and then you can get mimickers and reverser signatures. So this was a paper that, it was a seminal publication from 2006. And then the idea for Links was to really expand on this idea. So the Links cube is this concept of having many, many perturbations. Again, those could be chemical, genetics, microenvironments. And then you apply those perturbations to different cell types. And typically those are cell lines because those are easily manipulated for high throughput. And then you measure what happens to the cells before and after the perturbation with different phenotypic assays that could be omics assays or imaging or other types of assays that measure changes to what happened to those cells. So in the second phase of Links, so Links had 2 phases. The first one was 4 years. The second one was 6 years. In the second phase, the program was expanded to have 6 data generation centers and 1 DCIC, which was us. And this is the map of where they were on the US map. And this is represented on the portal that we developed for Links. As the data coordination center, and this will be a little bit relevant later, we are also a part of another common fund program called BD2K, Big Data to Knowledge. So we were serving on both sides. And the BD2K program was really about finding a solution to better manage data. And the CFDE is sort of like, I guess, a grandchild of that program. So one of the things that happened to us as the DCIC for Links, we were awarded at the same time that those data generation centers were awarded, and we had some time that we needed to wait for the data to be generated. So during that time, we really decided that instead of waiting, we're going to be able to just go to the public domain and create signatures from a lot of those publicly available resources like Geo and ENCODE. And so basically what we've done, we wrote code scripts that takes this data and abstract it into signatures. And those could be gene sets. And those gene sets can be converted to what's now called knowledge graphs. So connecting the genes based on their shared attributes, or connecting the attributes based on their shared genes, as well as this concept of bipartite graphs. So you have the attributes on one side and the genes on the other side. So we've been doing that systematically across many resources. And this was a really successful effort. So one of the two bioinformatics tools that came out of this are Enricher and Harmonisome. So far, we process data from over 100 different resources. And Enricher currently has almost 500,000 annotated gene sets. And you can see here that there is now actually 72 million submission to the system. So this is a screenshot from a few weeks ago. And then over the last 10 years, these two websites together saw at least 2 million unique users. So those are really turned out to be very useful resources to the biomedical community, and many of you probably have heard about these. So one of the things that we've done for links was we had to build all those portals as the data coordination center. So we created the information portal, which is at the linksproject.org, as well as we had our own center portal, which is at linksdcic.org. And we also came up with this concept of an app store. So instead of building a portal that has everything in it, we really build a collection of tools. And this is not just our team. This is a collective effort of the entire consortium, and different groups develop those databases and tools, and we all serve them in this links tool marketplace that currently has 69 different tools, and all of those tools are represented as cards. So this is sort of like was a revolutionary thing for bioinformatics, having all those tools in one place, and here they're sorted by the number of clicks on those cards. So this is just another view of all of those cards with all those apps. And this was really, again, very successful. Our team, the DCIC, published over 45 bioinformatics tools and databases. We had over 2.2 million unique users. And right now, we still have about 4,000 unique users per day, 45,000 unique users per month. And also those tools, many of them have become very popular and successful with over 15,000 citations. So several years after the program started, the second phase of the program started, the Broad came out with this another seminal paper that was published in Cell. And here they introduced the Links L1000 assay and data. And this is the L1000 assay is a high throughput transcriptomics assay that is an alternative to microarrays and RNA-seq. And it measures 1,000 genes, but the rest of the genes are inferred relatively accurately. So you get the full genome, and they were able to do this transcriptomic assay in high throughput. And in the publication, they showed how they had 5,000 genes, loss of function or gain of function, some biologics, and about 20,000 small molecules that were used. And those small molecules were used in different concentrations across many cell lines. And also the gene expression was measured at different time points. So this was like a massive expansion of the original data set, and now it's even larger. So there is almost all of the genes were knocked out with CRISPR, and the small molecule collection reached 30,000, and the total number of samples that were profiled at the end of the program was 4 million. So one of the things that we've doing, some of those apps that we developed, and this is I guess the L1000, you can say that this is the most substantial data set that was produced by Link, and also the most widely used resource that came out. So throughout the years, we developed search engines to query that data set, the L1000 signatures. So you come into those search engines with your data of, let's say, up and down genes, and then you get a ranked list of mimickers and reversers. And the most current app that we have for doing that is called SIGCOMM Links. It was published 2 years ago. And this is just a way that we were able to also make an impact by building those easily used search engines that were an alternative to what was provided by the Broad. And here is just one example of how those search engines have been used. So far, for those 4 tools that I just showed you, they were cited by about 800 papers, but many of those papers really are experimental validation of predicted drugs from those hypotheses that could be generated from those search engines. So this is one of the most successful implementations of this approach with our tool, and this project we were actually part of. And this was a collaboration with Chiara Giannarelli, that is now a professor at NYU. So Chiara, her idea was like to take PBMCs and then treat them with plasma of patients that have atherosclerosis, which is the clogging of the arteries. So in the plasma of those cells, there's some signals that promote the formation of foam cells. And what we wanted to find is small molecules that can potentially reverse that signature. And then one of the drugs that came out from this analysis, one of the reversers that came out from the querying of the RNA gene expression signatures from those stimulated PBMCs was seracatinib, which is an AstraZeneca drug that in cell-based assays, animal models, and specifically a rabbit model of atherosclerosis, showed like a significant reduction in plaque formation. So this project almost came all the way from concept to clinical trials. AstraZeneca decided that they do not want to proceed with clinical trials for this phenotype, for this drug, for some reasons, but we almost made it all the way through. And that was an exciting project that was published very recently, last year, in Nature Cardiovascular Research. So this is the SIGCOMM Links interface, where you come in with your up and down genes, and then you press submit, and then you get those ranked list of drugs. If you don't have up and down genes, you can start with a single gene or a search term like a disease, and then there's different ways that you can convert a search term into an input that then you can submit to SIGCOMM Links for getting those prioritized compounds. So the program has different sources of inputs, either a single gene, a gene set, up and down genes sets, or any search term. So the topic of this presentation today is really about a new method to query the links L1000. So if you think about it, there are about 1 million gene expression signatures in the database that we query. And when you get the results, the result could be overwhelming. And then there is this issue of having the very specific signatures for specific drugs in a specific condition. And one of the things that we are thinking is that we can create some type of a consensus approach to rank those drugs. So for example, if we have this drug, valdecoxib, many times in the returned results. So let's say if we're searching for compounds, there are 700,000, a little over 700,000 entries in the database. And then the drug in different cell types, conditions, concentrations is showing up in the results in many places. over the ranks. So what we want to see is the ranks of that drug concentrated at the beginning. So we can do it by looking at the ranks of the drug, or we can look at the score, whether it's a reverser or a mimicker, and what is the p-value or the z-score of the ranks. So we can compare that deviation from the expected rank or from the expected collection of scores, and then can prioritize the drugs now at the drug level, not at a specific condition. So just if I'm showing you here what the results right now in SYNCAMLIX look like, there is the drug name and then the cell types, the concentration, the batch, where it came from, and then the time point. So this is going to be potentially replaced by this consensus approach, or alternative to this, you can have this consensus approach that finds the AUC of the drug across all of the instances of it appearing in the results. And then there's going to be a p-value that is computed by the KS test and all of the conditions that were identified for that drug. And recently we applied this approach to a collaboration with two investigators from the University of Wisconsin, Mark Keller and Alan Atty, who are studying diabetes. And they're using this diversity outbred mice as a model for human disease. So there's mice, they were collected from different parts of the world to create this collection of genetically diverse mice that now you can do all kind of like creative experiments with. And they have 500 of those mice that are genetically diverse. And what they've done, they took out from those mice islets, and then they want to see how those islets are, how well those islets produce insulin. So they have applied different assays, first to profile the islets of their ability to produce insulin, as well as they did RNA sequencing of those islets. So for each mice, they have a sample of RNA-seq from those 500 diverse mice. And then what you can do, you can look at the insulin secretion. So this is a visualization of the insulin secretion for across the mice. So the mice are the columns, and the level of insulin secretions are the rows. And then you can see that there is a range of for different mice based on their genetic background. They're more or less responsive to secreting insulin. And then you can take the RNA-seq data, and then you can see how individual genes, based on their expression, correlate with insulin secretion. So now, instead of having like differential expression signatures, we have genes that are highly positively or negatively correlated with insulin secretion. And this is just showing that this gene E4B4 is highly correlated with insulin secretion. So at least for many of those known genes, you can see that they come in either on those edges of this ranking based on this correlation. And then what you can do, you can submit it to SYNCAMLIX, and we use this new approach, this consensus signatures KS test approach. And to test those drugs, they also needed a positive control. So this is a drug called ISX9 that is known to, when you treat the islets with this drug, they will produce insulin. And then this is the results of the screen. So they tested about 30 drugs, and you can see that we're really enriching for compounds, and there's several compounds that are doing much better than our control compound in mouse islets. And then the next step is now they're testing it in animal models as well as in human islets that are isolated to see if this also translates to human studies. So now I'm going to switch gears a little bit, and I'm going to talk about the CFDE. And the history of CFDE, like I mentioned before, it started with BD2K, and then there was this program called the Data Common Pilot Phase Consortium, the CPPC, and then the CFDE came after that. And then throughout that time, we were thinking a lot about finding solutions to data management, data analysis, and how to do those bioinformatics analysis in a better way. And we have this sort of like timeline of publishing those different tools that sort of like have some type of succession and were developed as we improved our understanding of how to solve those issues. So in 2016, we published this paper that is actually a Jupyter notebook. And this is in this journal called F1000 Research. And this is the RNA-seq pipeline that did some analysis on Zika virus. And the idea was that the paper is coming out with a Jupyter notebook that contained the paper in it. And then readers of the paper can take that paper and swap the input data and have their own RNA-seq pipeline analysis with all of the figures and results based on their work, on their signatures. So this paper sort of got some attention, and then several years later, there was a successor to this paper called BioJupys. And BioJupys really took advantage of this concept of Jupyter notebooks and some of those Jupyter notebooks libraries that can automatically execute the notebook. So with BioJupys, you can come in with your RNA-seq data, you upload it. It can be FASTQ files, or it can be a data matrix with your data. Then you have to make some choices of the tools that you want. You have to select your perturbations and control samples. And then you press submit to get a report that looks almost like a paper with your RNA-seq analysis. And that tool is still widely used by the community, and it's been a really great success. But then Daniel Clark, who joined the lab at that time, thought of even a better way to sort of like generalize this. And this when Apiters came. So Apiters was a successor of BioJupys, and Apiters is not really a bioinformatics tool. It's sort of like a way to develop tools in a more sustainable and easy way. So many of those Apiters can replace some of those bioinformatics tools that I showed you that we developed for links. And the way Apiters work, you start in with a Jupyter notebook, and then you annotate your notebook in a way that you can convert the notebook into a full stack web-based application that has like a input form. You upload your data, you set up some parameters, and then you execute the notebook in the cloud by pressing submit. And then you get those reports that again look like publications. So far we published over 100 of those Apiters, and all of those are served on this Apiters catalog, and they're still widely used. But then the next thing that we've done was to develop this sort of like the next step from Apiter, or the successor of Apiter, was this concept of the Playbook Workflow Builder. And this is a CFDE partnership that, again, led by Daniel's work on, coming up with this idea of taking the APIs from the various Common Fund programs, as well as many other sources and tools, and creating this network of microservices that can be chained to build workflows. So this is the graphical user interface workflow, bioinformatics workflow engine that I would like to quickly demo. So let me switch. So this is the website. and I will post the URL shortly. And here you can come in and you see all those cards, and those cards tell you what you can start with. So for example, you can start with a gene, and then here I can type in STAT3, for example, which is like one of my favorite genes. And then I'm presenting with another set of cards of what can I do with this gene. And those are different next steps. And you can see the breadcrumbs at the top show me how I'm expanding things. So here I can click on this query GTEx for median tissue expression. And immediately I'm getting results. So this is actually happened in real time, where it went to the GTEx portal and it got the expression of that gene across tissues, and then press extend. And now I can visualize this, for example, as a bar plot. And this takes a minute, but here you get a bar plot with the tissue expression of the gene across the tissues. And then what you can do, you can switch here to report mode. And the report mode actually wrote a story of what happened. So we started by selecting STAT3, and then we queried against GTEx, and then we visualize it with a bar plot. And this is a report that was generated, and then we can export the report, and then we can change the workflow. So we can go back to the workflow mode, and we can continue to explore things, or we can, start again with another gene and change the gene and get that result. So this is still a work in progress. Let's switch back to our presentation. So overall, like as we are proceeding with, working on the CFDE data resources, there are several apps that are emerging. We haven't stopped collecting signatures and converting resources into signatures, and we had this double drop of RamaGene and RamaGeo resources that you will hear about from Giacomo. And then everything is going to be served from the CFDE workbench, which also I can share the link from. And then there's some other apps that are sort of like in the works. So before I pass it to Giacomo to talk about RamaGeo, I would like to first thank the members of the lab. Daniel has been instrumental for working on the second part of the presentation, and Errol is also working on the links signatures part that I mentioned. And also you can see that we have a lot more faces on this picture plot, and this is because we have a summer program every year where we double the size of the lab, hosting 10 to 12 summer undergraduates, and they are starting on Monday again this year. I also like to acknowledge the CFDE team for their great support, and then Shankar for collaboration, and his team for collaboration on the DRC, as well as our collaborator at Wisconsin and NYU for those experimental projects that I mentioned, and also for the future collaborations with the Knowledge Center, and for organizing this seminar, as well as the partners, the Playbook Workflow Builder partnership that includes other members of the CFDE, as well as our membership in the Data Distillery Knowledge Graph partnership, which I did not talk about today.